blooms all across the northern temperate zone of the world. So here in Northern Europe, all throughout northern the US, and it's actually a global problem, although we all are experiencing it locally here in New York, where we have such a vast amount of water resources. And I know a lot of you guys are probably on Quagga Lake, and that's had a lot of algal blooms recently as well. So I'll focus a bit on some of the Finger Lakes in Cortland and Onondaga, and also Cayuga County, and some general issues as well. So a lot of you may or may not, I don't really remember personally, but I've learned about, and some of you may have family members who are older or may remember yourselves, when in the 1960s and early 70s, there was a lot of concern because there were fish die-offs and lakes were getting greener, about what was causing these fish kills and algal blooms. So the term algae is a term for microscopic photosynthetic things that live in the water. And so people were really concerned because obviously they're going out to the lake and fish are washing ashore, especially in the Great Lakes. And everyone was concerned that the lakes were dying, which is sort of ironic because it was sort of an excess of growth, not things dying, that was causing other things to die. And there was a lot of controversy at the time about what was causing this. And until recently, this is something that when I teach limnology, the study of lakes was considered something we all figured out and was a success story of limnology, because what was done back then, I would say it's like your grandfather's algal blooms, like those things, we sort of figured out what was causing them and helped to work to reduce them. And then in the past decade or so, that seems to be reversing. So what's going on? So back then, we had a bunch of scientists, say Dave Schindler, working up at this area in Canada, and a lot of scientists in the US did a bunch of experiments, some of which were pretty dramatic, where they would add various kinds of nutrients to lakes, and change characteristics of lakes and see what would cause growth. At that point, fertilizer and detergent people were saying it couldn't be phosphorus and they didn't want to change. And what people found is if they added nitrogen and iron and other things to the water, and carbon, those were not causing blooms. And then if you cut a lake in half or took another lake and added just phosphorus to it, even a small amount of phosphorus addition would make algae grow really, really quickly, because that's the limiting nutrient for a lot of phytoplankton. So you add it just like you would fertilize your lawn. If you add phosphorus to the lake, that's what's limiting the growth of a lot of phytoplankton. And so that was pretty established science back in the late 60s, early 70s, led to a lot of the regulations we have reducing the types of fertilizers and when you can put them on the landscape, and also getting better sewage treatment. And that helped to remediate our waters. The Clean Water Act went in and helped to make algal blooms less of a problem for a while in the 80s and 90s. So a bloom of algae sounds nice, like a bloom like a flower, but it's actually this growth, excess growth of these small photosynthetic things. And that caused this increased growth of them. So what we realized then in the 70s, or the scientists who were doing this realized, is that the phosphorus inputs were causing this, and those reductions reduced the algal blooms. And we sort of thought that was solved. It was taught as a success story, as I said, in limnology for years. And then in recent years, these algal blooms have been returning past decade. And my screen is not advancing. Hold on, there we go. So a bloom is a rapid growth of phytoplankton, a lot of biomass, so a lot of material there, just like if you have a lot of crop on the landscape. Not all blooms are toxic. So these days we are really concerned about harmful algal blooms or toxic blooms. In those days, we didn't study the blooms as much for toxins. So some of them almost certainly were toxin, but not all blooms are toxic. Some strains of these and some species of these phytoplankton make toxins, some don't. And a high growth of these phytoplankton can turn the water different colors. And those of you guys who have been on lakes a lot might have seen that it might be a green color, a blue-green color, even bluish or brown or red. That's due to different kinds of phytoplankton dominating the lake, getting really abundant. And they have different kinds of pigments, just like plants may be slightly different colors. They have different pigments that are capturing different wavelengths of the sun to help them grow. So by doing things to the environment that alter what kind of these little tiny photosynthetic organisms are growing, we can change the color of the water and also maybe be promoting sometimes the kinds of phytoplankton that are causing some of the harmful algal blooms that we're concerned about today. So it's really hard for us. We're like fairly big terrestrial land-dwelling organisms, and we're used to thinking of plants as our photosynthesizers that are pretty big organisms that we grow and eat. It's hard for us to like contemplate that there's so many different kinds of little tiny photosynthetic organisms living in our water and how different they are from each other. So for class, I sort of made a diagram to try to get at that. And so this is the tree of life based on genetic evidence, how different organisms are from each other. And basically, there's three big groups, domains of life. Some have complicated cells that are called eukaryotes. Some are bacteria. There's two big kinds of bacteria. And so you and I and our dogs and our cows and everything are in this complicated cell group. And so if we put a little arrow here, every kind of plant that you might eat or you might grow if you're a gardener or a farmer is in this closely related clade or group here of land plants. So the grass in this picture, this tree that was outside of the apartment I was living in when I was out in Santa Barbara for a year, all those things are in there, everything that you grow in your garden or your window pane. And then for our dogs and all of our agricultural animals, every animal you know is in this animal group here. And like I say in class, you would not confuse my dog or a cow or a cat for a plant. At least I hope not. You know that they're really different. So if the things that we're looking at in the water were as obvious as these big things we're used to, it may be more clear that phytoplankton are really different, because phytoplankton is not one group of organisms. It's a really distantly related group of things. So I'm going to put an arrow now in every group that contains organisms that are phytoplankton, so little photosynthetic organisms living in our lakes. And so you can see some of them are bacteria, like you might have heard of cyanobacteria or blue-green algae. There's also all kinds of eukaryotic ones that are more distantly related to each other than a dog is to a tree. So if we're doing things to the lake, changing nutrients, changing temperatures, other conditions, and we favor one of these things versus another, we can go from having a kind of little photosynthetic microscopic organism that's really nutritious and feeding small animals that then feed small fish and give you a healthy lake, and one that is favoring something that might be more toxic or have other deleterious effects on your lake. And so it can be really hard to relate to that because we're just large and we can't see that. If we were as small as they are, we know they were really different. But the things that we do to lakes directly, like changing nutrients and other things that are happening because of changes in the environment, can change this balance. And that can help promote things that we don't want to have happen, like promote harmful algal blooms. So these differences matter in lake ecology. They determine whether the algae are edible to the small plankton, small zooplankton, small animals that are eaten by fish. They determine if they're toxic or not toxic, if they have lots of nutritious characteristics, or if they're sort of junk food that's not very nutritious for the organisms in the lake. So if we were little, like all of these pictures that are different kinds of phytoplankton, that looks really different when they're blown up. But to you, if you're at the lake, it'll just look like, oh, it's a sort of blue or green color or a browner color on average, and there's hundreds of different kinds of phytoplankton present in the lake at any given time. So it matters because many of the possible factors that we have happening now are once again favoring cyanobacteria, and some are toxic. So some of the ecology in this of why the cyanobacteria are favored, we understood from way back in that time of the 60s and 70s when people were doing the first work. And some is new, some are new factors that may be occurring. So what we know from the 70s is that in most cases that phosphorus, like I showed you in those dramatic pictures of lakes being, having nutrients added to them, that phosphorus was the thing that limited their growth. You add more phosphorus and boom, you get these blooms. So we increase phosphorus loading by increasing septic input, increasing certain kinds of fertilizers. That causes this increased growth and this eutrophication, this excess growth. And so also it tends to favor cyanobacteria. Bacteria themselves have a different genetic structure than the eukaryotic cells. They require more phosphorus to grow well. They have a higher phosphorus requirement. And so that also often favors cyanobacteria because they're even more limited by phosphorus than other kinds of phytoplankton. And also some of the cyanobacteria can get another limiting nutrient, nitrogen, out of the atmosphere. They can break like a triple bond in the nitrogen that's floating around the atmosphere and is over 70% of our atmosphere, and make biomass with that. And other phytoplankton can't do that. So the more that nitrogen becomes limiting because we're adding more phosphorus, that also favors these cyanobacteria because they can get at atmospheric nitrogen. And we're also finding that a lot of the strains that are doing well now can also get organic nitrogen better than the other phytoplankton. So if you have urea and other things running into the lake, that can favor them. So the probability of having algal blooms as you get increases in phosphorus, it gets higher and higher. And so we lowered the phosphorus inputs in the last few years due to good land management, working with farmers, working with people not to apply fertilizers to their lawn, having better requirements for sewage treatment. And that reduced algal blooms in many lakes. And so that was a real success story around the globe. We had these predictable responses. We knew for a certain amount of phosphorus, we'd get a certain amount of algae. And what's happening in some of the lakes that you guys are interested in, especially some of these finger lakes, is that even without a huge increase in phosphorus in these systems, we're getting these blooms and sometimes harmful blooms. Increasingly, the DEC has been great about having the ability to get samples analyzed quickly for toxins. And since they've been doing that during this last decade, there's lots of increases in harmful algal bloom and toxic algal bloom reports. And then that's of concern, not just because of taste and odor issues and potential problems, but actual problems with toxins in the water that might be harmful if you're drinking that, and also might be harmful to swimmers or dogs that go in the water and then lick their fur, and to wildlife as well. So we're seeing this increase in cyanobacterial blooms again, including these harmful algal blooms, which are ones that we now get tested to be toxic, even in some lakes that have relatively low phosphorus concentrations where you would not be expecting, in theory, based on what we know from the 70s and 80s, that you would get algal blooms. So this is not just true in central New York, but it's also true around, like I said, northern US, northern Europe, even in the Arctic systems, where we're getting these blooms. So what else has changed? Like, why are these cyanobacteria blooms increasing? And so one of the things is just the temperature itself. Warm temperatures tend to favor cyanobacteria. There's lots of different things that we're doing that are creating conditions that favor these cyanobacteria. So in lakes, we're seeing the effect of increased temperature around the globe, especially in northern zones, faster than we are in the atmosphere. And part of that is because we're seeing changes in the amount of time ice is on a lake. And you guys may have noticed this living around lakes. And people back in the olden days, in the 1700s, even 1500s, people would write in notebooks like when the ice went on and off a lake. They didn't have other entertainment back then, and in their diaries they would write down the ice came on this lake this year, went off that year. And in the States, we tend to have written records from the 17 or 1800s, but in Europe they go back even further when people were writing this down, and you can record and see the times that ice went onto a lake in the fall and ice came off the lake in the spring. And we've lost, say around here, 2 to 3 weeks of ice cover on average. And some of you may have noticed that if you're ice fishermen, it's less likely to get enough ice that you can go out and fish every year. The ice comes off earlier. And so if you, say, fought two of those different kind of phytoplankton against each other with the same kinds of nutrients, but you raise the temperature just a small amount, a couple of degrees, you would end up having cyanobacteria win competition for nutrients. The other thing that makes that ice off really important is you might have noticed when you're at a lake in the summer, if you're swimming or your kids are swimming, if they dive down or if you dove down when you were a kid, the surface part is warm, right? Because once the ice comes out, the surface water gets heated by the sun and becomes nice and warm and you can swim in it. But below that, the warm water is less dense because all the water molecules, when they're heated, are moving faster and it makes it less dense, like makes them further apart. Warmer water is like floating on top of the colder water below it, just like oil would float on water in your salad dressing or in vinegar in your salad dressing. And so when you swim down from the surface in the summer, you'll see this really nice shallow mixed layer like you could see here, where all the wind action is occurring and you're getting the shallow warm area where you're swimming at the surface. And then below that, it stays cold. All that heat energy from the sun gets put into that shallow layer. So the sooner you get ice off and the sooner you make that warm layer at the surface, the more that can heat. So we're even seeing in lakes like Lake Superior up in the far north of the US and Canadian border that has lost on average a few weeks of ice a year, and the temperatures there in that layer where all of the energy is concentrated, that ends up getting warmer. So making the water warmer also can favor these cyanobacteria, these big climate effects. In addition, because we have that warm layer floating on top of the cold layer, you get sort of not just waves at the surface, but that makes waves at the boundary, like you can see in this image, between the bottom of that warm surface layer and the cold layer below it. And just like the waves at the surface break on the shore, sort of crest and break at the shore and stir up sediments, the waves at the bottom of that warm surface layer break at the bottom. Like you can see here, it's called an internal wave because it's not at the surface, it's internal to the lake. And that breaks at the shoreline. And that can stir up sediments. And sediments are also really rich sources of nutrients. Just like your soil is a rich source of nutrient for a plant, the sediments are a really rich source of nutrients because everything that was living in the lake and died, all the phytoplankton and the zooplankton, the fish, sinks down to the bottom and ends up in the sediments decomposing, and those nutrients can be returned into the water when that breaks. And all of the nutrients that are in there from times when farming practices say weren't as good and people came and clear cut the landscape when people were first settling, all of those things that went into the sediments originally when we had bad land management practices are still in the sediments, and all those nutrients are there. If you're stirring them up, that can add extra nutrients to the lake. So we can call that like the legacy phosphorus from those previous times can be added. So even if we're not adding a lot of nutrients to the water now, that legacy phosphorus is still in the sediments, sort of ready to be put back in the lake and make algal blooms. And this has been studied really well by Nelson Harston's group at Owasco Lake and by a number of investigators at Skaneateles Lake. So in addition, just like you might have different optimal fertilizers for different kinds of plants, the nitrogen and phosphorus ratios that are coming into lakes will affect what type of those different phytoplankton you're favoring. And also whether you're having inorganic nitrogen like nitrate, or organic nitrogen like urea or stuff that would come from animal waste. And organic nitrogen tends to favor cyanobacteria, and that's in some fertilizers now as well. And another possible issue would be invasive mussels. We've had zebra mussels and quagga mussels that have come into the lakes over the past 20 to 30 years. And there's some thought that they may also be promoting the nutrients and conditions that favor cyanobacteria. And this is true in many parts of the world. These have invaded Northern Europe. They're originally more from Central Europe. And so it's not just here that we have these invasive mussels. They've invaded throughout the northeastern US and other parts of the world as well. So we first got zebra mussels in this area. If you've thought about them at all, it might be because you stepped on their shells and they cut your feet and they're nuisances. These very small mussels. A lot of times people sort of like them because they make the water clear. And so they think, oh, like I can see further. You know, one of the things I did when I was an undergrad was I had a job diving for invasive plants, and you know, there were not mussels in the lake right when I was there, and they've come since. And you know, now there's a whole diving industry, say up in the Thousand Islands, because it's clear now because of the mussels that wouldn't have existed before. So what happened was we had these like sort of eutrophic lakes from the 80s and so forth. Mussels came in, and the mussels filtered that water. They excreted, they basically, like us, they're animals, they release nutrients as excrement, and that goes right along the bottom next to them. It fertilizes plants. So you might have also either yourself noticed or other people might have noticed that your lake has become weedier since maybe a few decades, 3 or 4 decades. Part of that is that the water is clearer, so those plants need light, so the light is penetrating deeper to grow plants, and also the mussels are excreting and fertilizing the sediment there where they're growing. So we have this benthified or bottom-driven system as opposed to an open water system. And so we get high light, high phosphorus near shore from the excretion of the mussels, and we have lower offshore phosphorus levels due to regulation and nutrient release than often near shore. So we can have near shore pockets with really high nutrients above those zebra mussel beds when we just had these zebra mussels present, and they were present on hard surfaces, mostly near shore. And that also, especially starting in like the 80s and 90s, late 80s and early 90s, started growing higher growth of slimy filamentous algae and pond weeds. And there's another reason that this might help cyanobacteria specifically is that you might think a mussel is just a dumb little invertebrate, doesn't even have an obvious head, right? But when they filter, they sort of suction the water that has all those different phytoplankton in it. They can taste and they can use various sensory mechanisms to ingest some and release others. And when they release them, they are often released in these sort of pseudo-feces in these nutrient-rich areas. And so the things that are distasteful to them, they don't eat. They're not just blindly filtering. And one of the things that they tend not to like are some kinds of cyanobacteria, these toxic blue-green algae. So they're eating a lot of the algae, but they're not eating a lot of these toxic algae. They can reject them. And that's been studied by lots of people. Ace Sarnell at Michigan State did some really great work on that. And so the first mussel was a zebra mussel. You may have seen these like little algae blooms of filamentous algae near shore. Those aren't toxic, but they were nuisances. No one wants to walk through all this scum to go swimming in their lake, and it rots on your beach. So you've got mussel shells, and then you've got this cladophora, this green algae bloom. But it's not toxic. It's unpleasant, unsightly, might cause people to not want to go out there, have to wear water shoes. And then we got quagga mussels, a second kind of mussel in most of the Finger Lakes and a lot of inland lakes. They're not everywhere yet. And the quagga mussel is a relative of the zebra mussel. It's a little bigger, and it can live on soft surfaces. So it's not just restricted to the rocky areas. It can cover a lot of the bottom of lakes, and it's tolerant of lower temperatures, and it's a little bigger, and generally it can eat more and also sort of select more than the zebra mussels can. So we're not just covering a bathtub ring around the lake with mussels anymore. We can be covering most of the bottom of the lake with mussels now. So if you look in the different lakes we're talking about, you can see the timing of when the zebra mussels and quagga mussels came in. Zebra mussels much earlier than quagga mussels. Otisco apparently just got quagga mussels reported last year by Lou McCaffrey at DEC. So they're rejecting, you can see these little siphons on this picture. They siphon things in, they reject them out. And so that can increase the toxic algae. Also, they're animals, so just like you and me, right? We're eating food and then we're excreting waste. And they excrete waste, and they can alter the amount of nitrogen and phosphorus that's in the bottom and fertilize everything. And they're also all along the bottom, and they're animals. So just like you and me, they're using oxygen out of the water. And when they use oxygen out of the water, they make less oxygen at the sediment water interface. And we'll talk about this again in a little bit, but when you have low oxygen at the sediment water interface, one thing that can happen is it can favor the release of phosphorus out of the sediments. So Charlie Driscoll and some other people Charlie Driscoll at Syracuse University are trying to see if maybe just the presence of the mussels and the low oxygen concentrations they may be making may also be favoring release of this historically laid down phosphorus in the sediments. Another reason this could favor our toxic algae, unfortunately, it's like a whole suite, a perfect storm of conditions, is that the zebra and the quagga mussels are at the bottom, and we see that the cyanobacteria, many of them can control their buoyancy. They don't have flagella, they can't swim like some of the other phytoplankton can. However, they are able to basically inflate little structures inside of them that act as a buoy that bring them to the surface and then deflate them so they can go to the bottom. And so they often, and this is a great study by Kathy Cottingham who first really quantified this, but they can move down at night and then go to the sediments where these nutrients might be releasing and the zebra mussels and quagga mussels are excreting. They can suck up nutrients, and then they can go up in the day to photosynthesize. So whereas all the other kinds of phytoplankton that aren't toxic and so forth are stuck in the surface water in that warm surface layer, these guys are able to float and then sink down and then come to the surface. And we've seen that while diving sometimes. We go out to collect samples and you'll see that it's clear in the morning, and then you're diving down, it gets really murky at the bottom, and then as the day goes on, you see that murky layer raising. So by 8 or 9 o'clock, you realize like, oh, you were diving through this layer of toxic algae that had been at the bottom at night and was coming to the surface at the day. Now that we know that, we sort of abort the mission when we see that. But until then, it was sort of a shocker. So they may have access to this excreted nutrients from the mussels or extra nutrients from the bottom. And so that could be an important factor as well. So the little bit of work that we did on this, working with Upstate Freshwater Institute, was to follow up on a study, a really nice study that was done in Owasco Lake by some researchers from the Cornell Biological Field Station, Jim Watkins and Lars Rudstrom and Ed Mills and Mark Leopold, as well as some researchers from Finger Lakes Institute and USGS. And they had gone out with this thing called a Ponar grab, and they were able to then grab samples from the bottom and count the number of mussels that they had. And they did this in 2007 just to see what was happening and to see what mussels were there. And they sampled along Owasco Lake on a north-south gradient, 10 different sites, and at 10-meter depth intervals twice in June and October. And at that time in 2007, they found zebra mussels present, but none of the quagga mussels that could live on hard surfaces and tend to be able to live deeper. And one of their main take-homes was that this was a big potential threat to the lake because there were a lot of important native invertebrates that they were finding at those deep sites that are important to fish that they worried would be inhibited by the presence of quagga mussels. So they found that the mussels, the zebra mussels, were generally much more abundant, shallower than 10 meters. They could extend down to 30 or 40 meters on the south end. And they were, where they were present, they could make up between 1/3 and 90% of the organisms living at the bottom of the lake. And so the native diversity was lower in the shallow sites with zebra mussels than in the deeper sites. So Upstate Freshwater Institute is making a model, a water quality model like the one they did for Cayuga Lake a few years ago. And they are parameterizing the model with some of the data that we collected for mussels and plankton. And I'm trying to make this advance. And so in Cayuga Lake, they found that the mussels themselves were responsible for increasing phosphorus, perhaps 20 to 30%. And they want to do the same estimates for Owasco Lake. And they've made a Owasco Lake model that's using these mussel estimates. And so we also wanted to know for sure had quaggas arrived in Owasco, because we thought they had, but that had not been officially counted. Lou McCaffrey had been doing some images, so we also took images to try to see if that would be an easier way to quantify the mussels. Lou McCaffrey from DEC That's been pioneered in marine systems where you can just take pictures and count things in the pictures. So we took images, and that could potentially allow a lot more faster estimate of spatial patterns. And you might be able to get computer learning to do that faster. So we went out, we had a structure that had a GoPro. You're looking through the GoPro right now. I'm looking at the 1/2 meter by 1/2 meter quadrat. And we would lower those down. It took pictures all the way down and then pictures while it was at the bottom. At the site, we collected nutrients that UFI would measure. And then we also collected the mussels from the quadrats once it was down there. There was a tender on the boat. 2 divers went down the line. The first one, before disturbing anything, took the nutrient sample. The second one collected all the mussels, and we would switch those jobs at either site. And Upstate Freshwater Institute analyzed the bottles, and we collected the mussels and in these mesh bags, and it took much longer than we thought to count them because there were tons of them out there. And so they were in a cooler, and then they were frozen until analysis, and a little army of ESF students and myself counted a bunch of these. So we gave the water to UFI that same day. That was analyzed immediately to see what was being excreted over them, so they could use that in their model. We stored the image from the quadrats, and we collected these mussels and analyzed the total number, if they had been alive or just were dead shells, because that could mess up the images. There's a lot of dead shells out there. And if they were Quagga mussels, the new ones that invaded that can go deeper, or zebra mussels. And we also needed to measure the sizes so that Upstate Freshwater Institute could estimate how much they're eating, because little ones eat less than big ones. So these are the sampling sites. We did a bunch of different transects from shallow to deeper. And some of these sides of the shoreline are steep, so those are why the dots are close together. At the north end and south end, there were two transects, and then one east on the east shore and one on the west shore. And on the way down, sometimes they catch something like a fish swimming by, but this is what the bottom of the lake looks like. A bunch of mussels down there. And so we collected those and counted them, and very laborious. There were lots more mussels on the previous survey, as you'll see. So here's zebra mussels in the sort of orangey color, Quagga mussels in the blue, and the total density of mussels in the gray. And we're going from 0 to 25, actually 23 meters deep. And you can see that there are a lot more mussels, a lot more Quagga mussels and zebra mussels at everything except the really shallow sites. And there are sometimes about 30,000 mussels in a square meter, so about a square yard, a little more than a square yard, 30,000 mussels in that amount of space. So imagine it's a huge amount of mussels that are present at the bottom of this lake. Imagine 30,000 mouths in a square yard that are filtering the water and excreting material into the water. And so if we compare our values, here's 5,000, which was the high point here in the Watkins survey. You can see the zebra mussels, which was all that was present in the lake then, are not too much more abundant than he found. But the Quagga mussels really increased that number of mussels that are there. The zebra mussels haven't changed that much, but they've been overwhelmed by the amount of Quagga mussels in deeper areas, and there's huge amounts of Quagga mussels out there. We also sampled some plants, which you can't take very good pictures of and can't estimate on the plants because all you see is this green. Images don't work there, and it's laborious to move them, but you can get plants in the lake, depends on the light penetration in your lake, but they'll go down to about 4 meters in Owasco Lake, depending on the light penetration. And on these plants, there was a range between like 500 and 7,000 mussels that you'd find in a meter squared on the plants. So that's a lot of mussels that are hanging out on plants. They can move, they can drop off the plants after the plants are dying off and go down to the bottom and live. We tend to think of mussels as stationary, but as you'll see in a minute, they're not really that stationary. So you can see the deeper you got, there were fewer plants, but there were more mussels on the plants. In shallow waters, a lot of times mussels get scraped by ice in the winter, and that removes them, and they all have to resettle from new ones. So that's part of what drives this shallow water versus deeper water pattern too. So we actually have counted everything. The numbers are high and variable. Some are small mussels. Larval mussels seem to recruit to plants, and they can move. And if you think about them recruiting to plants, it's sort of like blue mussels that you might have eaten in the store. They are on the plants, in the water, not just stuck eating things that are at the bottom, and phytoplankton that are in the water that's at the very bottom, but they can eat phytoplankton that are in the whole water column then, and maybe change the composition based on what they're eating and excreting, even near shore in those plant beds. And they're also promoting plant beds by making the water clearer and increasing the plants. So when we think about how a lot of people find the plants near shore to be nuisances when they're in high abundances, these mussels are making the water clearer and maybe also fertilizing the plants. It's sort of more like an aquaculture facility there for more mussels. So we also found a weird thing, which is at the deep sites when we first went down, when we put our hands in and we were just collecting the mussels at the surface, we found mussels deeper and they seemed to be closed and perhaps alive. So it wasn't part of the grant, but we actually took 3 sites where we took a smaller quadrat and just sampled down to, you could actually, the composition of that deep sediment was basically, I could stick my whole arm up to the shoulder into the sediment. That's how like unconsolidated it was. So the other diver and I at 3 sites collected the mussels in a smaller known area and measured the amount of subsurface mussels that were present. And if you remember, zebra mussels, usually these are almost all quagga mussels, just there'd be 1 or 2 out of 5,000 that were zebra mussels in these deep sites. And you can see here the deep in the sediment, not at the surface, which is what we were counting and thinking was alive and comparing to the images. Deep in the sediments, we actually would find over 5,000 of those living quagga mussels. Now, whether they're dormant, if it's a one-way ticket down, if they can make it back up, we are not sure. But that's interesting too, because they're still respiring then in the sediments if they're alive, and that might be also reducing the oxygen concentrations and releasing more phosphorus. So about most of the mussels living deep were quagga mussels. About 14% overall, those 3 samples were zebra mussels. They averaged over 5,000 per meter squared. So remember, that was the high concentration at the surface in the survey that Watkins and others did, like in the lake in 2007. So there's that many living ones in the deep sediments, and we had no idea they were there. Haven't seen that reported anywhere else. And it was about 20% of the mussel density at the surface. So 1/5 of the population is living below the surface. So we don't know, like, what is the implication of this, of these deep mussels? Are they helping like an earthworm to stir this up and release sediments, changing the oxygen? What's happening? It's a really unknown question. We never would have noticed this if we just took those grab samples. So if we hadn't stuck our hands in the sediment and realized that there was stuff down there. So that can show you the importance of different types of observation. Before COVID-19, I had a undergrad, Jack Zhang, who was looking at motion trails, and people have reported that mussels have moved before. They can move quite a bit, like 20 or 30 centimeters in a day. This one moved across the tank in less than a day, 18 hours. And he also noticed that some of them moved deeper. Then he had to leave school a few months early, so we don't know if they can bury themselves and come back up, if that's a one-way trip, but that's a really interesting thing for someone to follow up on. Again, there's been a lot of work on the idea that mussels can move horizontally on the surface, which is interesting to most people. Don't think of mussels as moving, they use their little foot to move. But burying is something we haven't really thought about. A lot of the mussels, other mussels that people observe, live on hard substrates. So an experimental approach might help, like has been done with zebra mussels. How much are they excreting? What's happening in lake and out of the lake? And right now, Charlie Driscoll and Chris Schulz over at Syracuse University, along with their technicians and a graduate student, are doing a sediment survey and flux study. And they're out there with chambers trying to determine like how much nutrients are being released relative to different kinds of sediment and whether mussels are there. and follow up on this related to the type of sediment that's present and if that legacy phosphorus is being released. So hopefully they're getting results right now. They're out there right now, and hopefully they'll know a little more how that's working by next year. So I'm interested also in what about those deep mussels? Are they active? You know, what's going on? Is that widespread? And is that also important? Just like earthworms are important for mixing soils, are they actually now serving to mix lake sediments that haven't been mixed? We didn't have something that was doing that as deeply. So it's unclear. And then also the role of plants as maybe causing this selective feeding by mussels in the nearshore zone. So I'd say we have this issue of how are we going to control these harmful algal blooms and just excess phytoplankton in general in this new normal, which has to include more than just phosphorus control. So when we fertilize with urea and organic materials, that cyanobacteria like, that can then favor cyanobacteria. We have these excess warm waters, we have less ice on the lakes, so maybe we need to even have increased nutrient remediation to make up for that. Maybe we need to think of other ways to change the balance of competition. And we need to think about, you know, preventing, if you can prevent mussels from leaving your lake and going to the next lake and exacerbating this problem, clean, drain, and dry your boats. That will help them not get these issues, but also what can we do to maybe moderate or reduce the impacts of these invasive mussels and contributing to this problem, nutrient recycling, and maybe changes at the sediment water interface. And when we have more storms that are concentrated, that can change runoff patterns too and change the amount of nutrients that are coming into the lake. So we have to have a holistic approach. Like, we may need many solutions, many small solutions to combat this. It may not be as simple, I don't think it will be as simple as just reducing phosphorus anymore. We have so many pressures now that are pushing the system toward cyanobacteria and toward harmful algal blooms that we need a bunch of small solutions to counter those big pressures. So we need multiple innovations. There's a chance for lots of ideas of how to make these conditions for HABs less likely. It's not easy not to have a simple solution, but it offers opportunities, right? We have all this legacy phosphorus in the sediments, but we also have a phosphorus shortage. You know, in agriculture, we're running out of the kinds of rocks that are bearing phosphorus for fertilizers. If we could take phosphorus somehow efficiently out of sediments and recycle that, then that might also reduce the source from the lake and provide benefit to agriculture if we could harvest that legacy phosphorus. So all these things we have to think about in the way limnologists will work with other managers to solve these problems, just like our forebears did solve these problems in the 70s and 80s. We're going to have to work together to solve these problems again in this century. So I'd just like to acknowledge sponsors and partners, and also all of the undergrads and assistants who helped with this, and Charlie Driscoll, who is great to talk with about this and doing some excellent follow-up research and provided that last slide. And I'm happy to answer any questions people have. I know it's late, and later than we expected it to be. So thanks for bearing with me. And if you're tired and want to email me